speaker is Shaelyn Silverman. Shaelyn is a recent graduate of the University of Colorado at Boulder, triple majored in molecular biology and neuroscience.
Science is filled with many intriguing questions, but perhaps the most profound and enigmatic of them all is the one that we've been asking since humans first looked up at the stars. Are we alone in the universe? And now we're finally on the verge of being able to answer this question, thanks in large part to the Kepler Space Telescope, which discovered thousands of planets beyond our solar system. So here are a few of the planets that Kepler has discovered so far. These are all roughly Earth-sized and may have liquid water on their surfaces, and so they could potentially be habitable. And there are a lot of other planets out there that are just like these. So the next step in our search for life is to determine whether any of these planets is habitable or inhabited. And this will largely be done by looking at their atmospheres. But we just have one problem, and it's that we only know of one habitable atmosphere in the entire universe, that of Earth. So that's a sample size of n = 1. That's a pretty poor data set, and unfortunately doesn't give us a lot of information. But we know that Earth has remained inhabited for the last 3.5 billion years. So what we can do is we can take atmospheric snapshots throughout Earth's history, throughout the time that it has been inhabited, and this will increase our catalog of known habitable atmospheres that we can then compare to all these exoplanets. So, sounds like a good plan, right? Well, we're not at a solution yet because this is still a pretty daunting task. I mean, taking the entire atmosphere and studying it over billions of years of history just doesn't seem too feasible at this point. So instead, what we can do to make this problem a little bit simpler is we can just focus on individual gases in the atmosphere and trace their evolution throughout history. So here I'm looking at nitrogen. Nitrogen is the dominant constituent of our atmosphere today. It makes up about 78% of the atmosphere. But it hasn't always remained 78%. In fact, we think that this amount may have drastically fluctuated throughout Earth's history, and we want to better understand how. But the ancient atmosphere is notoriously difficult to study because its physical properties, density and pressure, leave pretty imperceptible imprints in the rock record. And so, as it is, only a few geobarometers currently exist today. A geobarometer is an indicator in the rock record of atmospheric pressure. And actually, my mentor here, Sanjoy, pioneered early Earth geobarometry and invented 2 of the 3 techniques that we use today. And right now, we're currently trying to develop 2 more using cyanobacteria. So cyanobacteria, in my opinion, are the greatest microorganisms that have populated this planet. They go way back. They were actually the guys that released all the oxygen into our atmosphere. And so they're arguably the most important out there. But specifically here, I'm looking at how cyanobacteria record information about nitrogen pressure through a couple of different ways that I'll get into soon. But before I do so, I just want to give some background into the mechanisms that we're looking at here. So when cyanobacteria are deprived of biologically useful nitrogen like ammonia, they begin producing this protein called nitrogenase. Nitrogenase fixes nitrogen. So what that means is that it takes this atmospheric nitrogen and it converts it into ammonia. So that's a form of nitrogen that all these cells can then use. Now, nitrogenase is a very important protein, but the only problem with it is that it's poisoned by oxygen. So cyanobacteria then have to figure out a way to protect this protein from all the oxygen in the atmosphere. And they're very clever organisms. What they do is they develop these specialized cells called heterocysts that basically keep out all of the oxygen and allow nitrogenase to operate smoothly inside the cells. So what's happening here, I'm going to switch to this hand. What's happening here is that this atmospheric nitrogen is converted into ammonia in these heterocysts and then is laterally distributed down the filaments to the intervening vegetative cells that consume the ammonia as it passes by. And when this ammonia runs out somewhere along this filament, a cell will be starved of nitrogen and will be triggered to develop into a new heterocyst. So that's the mechanism that's occurring here. And we took this mechanism and we proposed the hypothesis that this distance between heterocysts should reflect the partial pressure of nitrogen in the atmosphere. So now I'm going to walk through how exactly we came up with this. So first of all, the distance that this ammonia should travel down this filament should scale pretty linearly with the rate of nitrogen fixation occurring in these heterocysts, which in turn should depend on the amount of nitrogen available to the cells, or in other words, the nitrogen partial pressure. So that's how we establish this potential hypothetical correlation between nitrogen partial pressure and heterocyst distance, in that if the nitrogen partial pressure increases, heterocyst distance will increase as well. Or so we wanted to see. So of course, we're scientists, we wanted to test this, and what we did was we cultivated cyanobacteria in artificial seawater that did not contain biologically useful nitrogen, so no ammonia in that seawater. And I grew the cells under these air spaces of variable nitrogen partial pressures. And then I ran those experiments for a couple weeks, and I looked at the heterocyst pattern that resulted. And now I'm going to show you guys some micrographs, a compilation of the pictures that I took of these cells to show you what I saw. So here, to explain this, going down these panels, we are increasing the nitrogen partial pressure that the cells experienced. And you can see that, so these arrows, they denote the successive heterocysts. You can see that as we increase the nitrogen partial pressure, this distance between heterocysts did in fact increase as well, which was really exciting because that's what we predicted, that's what we were looking for. And I was able to quantify this distance, or the relationship between heterocyst distance and nitrogen partial pressure. And we're now looking at potentially using this data as a possible Geo Barometer, because cyanobacteria are extant organisms, so they exist today, but they've also arguably been found fossilized in 2-billion-year-old rocks, such as this one here, and more convincingly found in more recent formations, and so could potentially reveal information about the nitrogen levels at the time they fossilized. And so that's where we are looking to take this research next, and I'm going to actually come back to this point, but before I do so, there's another part of this story that I want to tell you guys about, and it comes from the fact that this atmospheric nitrogen comes in 2 different isotopes. So an isotope is an element with the same number of protons but a different number of neutrons. So it's basically the same element but with different masses. And here, in this case, nitrogen-14 is the lighter isotope right here. And nitrogen-15 has 1 extra neutron right here, and so it's 1 mass unit heavier than nitrogen-14. And remember that protein from before, nitrogenase. Well, when nitrogenase is fixing nitrogen, it has a preferred ratio of isotopes that it incorporates. So under our current atmosphere with 78% nitrogen, this ratio seems to have more nitrogen-14 in it. And so the enzyme, or the protein in this case, nitrogenase, it prefers that lighter isotope. And I basically wanted to see whether altering the amount of nitrogen available to these cells would alter this ratio of isotopes incorporated during nitrogen fixation. So that's what I was looking for, and the way that I did this was, again, I grew my cells under variable nitrogen partial pressures, and then I put the cells in an instrument called a mass spectrometer, which gives isotope measurements, and I looked at the ratios that resulted. So before I show you guys the data on that, I just want to take a step back and make this a little bit more digestible, because isotopes, well, that's kind of a scary word. So instead of thinking about this mechanism in terms of isotopes, let's think about it in terms of beer, because I think that's a little bit more relatable to all of us. And instead of nitrogen-14, the isotope that nitrogenase prefers in this case, let's imagine a nice, cold, refreshing craft beer. And instead of nitrogen-15, that less preferred isotope here, maybe let's imagine a warm can of PBR. And as I mentioned, nitrogenase, it has a preferred ratio of isotopes that it incorporates during nitrogen fixation. So here, that ratio will be represented as a ratio of beer that we're using to fill a cooler. So now to extend this analogy a little bit further before I show the data to kind of help explain it more, imagine this. If you have less money, so maybe you're a broke college student trying to fill your cooler with beer, you're going to have to pick more PBR because that's the cheaper option in this case and you can't really afford anything more expensive. But now, let's say you have more money, maybe you have a job at this point, you'll be able to fill your cooler with more craft beer. You'll be able to be more selective and afford more of this craft beer. And in this case, the amount of money that we have is analogous to the amount of nitrogen available to these cells. And now I'm going to superimpose this on my isotope data to help explain that. So here is the data that I collected on my isotopes, and just to orient ourselves, I'm going to go through the axes really quickly. On the x-axis, we have nitrogen partial pressure, which in our beer analogy is the amount of money we have available overall. And on the y-axis, we have the nitrogen isotope ratio, measured in per mil, or parts per 1,000. And up here, these less negative values, that means more nitrogen 15 was incorporated into the product here, or in our beer terms, more of that PBR. And down here, more nitrogen, or these more negative values, that means more nitrogen 14 was assimilated, or in beer terms, more of that craft beer that we like more. And so you can see with this graph, there is a very clear, pretty linear trend between the two, nitrogen partial pressure and the ratio that resulted. And so just to kind of explain this in terms of the beer model, if you have more money overall, you'll be able to be more selective with your choice and choose more craft beer. So it's the same with the isotope case. Nitrogenase is able to be more selective when it has more nitrogen available to it, and it's able to incorporate more nitrogen 14, which is what it prefers. So I think that this model hopefully will help you remember what I found here with these isotopes. But there's something that's really exciting about this data, and it's that no one has ever shown this relationship before. So this is a pretty novel discovery in this field, and we're really excited about it, but even more excited about the fact that we might be able to use this as a Geo Barometer in addition to the Heterocysts one. Because nitrogen isotope ratios have been reported in sediments throughout all different time periods in Earth's history, but to our knowledge so far, no interpretation of these measurements has taken into consideration the fact that nitrogen pressure has changed over time. And so if we can apply this data to these measurements that are reported in the literature or that we find out there, then we can potentially measure ancient nitrogen pressure. And that's where we are looking to go next with our research. So as I've mentioned, the next steps are to apply this data now to the rock record. And I currently have some rock samples from the Gunflint Iron Formation in Ontario, Canada, which is up here, where this very famous microfossil came from. And what I want to do with these samples is I want to look for cyanobacteria fossils and see if I can apply my heterocyst and isotope data to these fossils to extrapolate information about ancient nitrogen pressure. And so that's where we're going next with this. And something really exciting happened on Monday. We actually received approval from Ontario Parks to go to the Gunflint Iron Formation and survey the locality. So we're really, really excited about that because the Gunflint is a very, very famous place. It has some of the best preserved microfossils of ancient Earth, and should be a lot of fun. It's a many, many geobiologists' dream to go there and explore. So very excited about that. But now to kind of summarize this talk and to wrap up, if we can measure ancient nitrogen pressure, then this will contribute to an overall better understanding of the ancient atmospheric composition and total pressure, which in turn will enable us to ultimately assess whether exoplanets are habitable based on their atmospheres. And so with that, I would like to thank everyone who has helped me with this research, especially Sanjoy. Thank you so much for everything you've done, and I'd like to acknowledge my funding sources. And if there's one thing that I would like you to take away from this talk, besides my incredible beer analogy, is that cyanobacteria may offer insight into Earth's ancient atmosphere. Thank you.